Hello everyone. Today in this video we'll be discussing about Python for bioinformatics. Before I start this video, let me tell you, if you are someone who's belonging to life science background or someone who have not done a single line of coding or not used Python before, you have come to the right video. You might be little overwhelmed by hearing Python. Someone belonging to life science background, we have not done any coding, right? But right now, in current scenario, industry and in research work, there is integration of AI ML. So there is necessity of integration of Python in biotechnology or in the field of pharmaceutical science. I will show how we can integrate this and we can carry out our day-to-day daily research work. There is no need to be overwhelmed. I will give you a roadmap and teach you the basics of Python. So before I start this, Python, just think him as your lab assistant. A lab assistant is someone who is very quick, who does all the work, someone who never gets tired. If you give him a command, he does exactly what you say. But the command must be precise, very accurate. If you tell him to bring a specific instrument or some equipments in the lab, he will bring and give it to you. We'll treat Python as your lab assistant. Just consider him as your lab assistant and I will show how we can integrate Python in our drug discovery, might be in clinical research or in biotechnology. Not just Python, there is integration of Python with several other libraries. Libraries means in the sense tools. There are separate Python tools for separate tasks. If calculation is needed in your data, we can use NumPy. If you want to visualize your data, you can use different tools. I will show you one by one and I will demonstrate how we can use Python in our managing our data, maybe separating out data. So manually, if we have a large number of data, it is very difficult to segregate or separate out data from that spreadsheet, right? Manually, it will takes a lot of time. If we use Python in segregating or if we separating out data from our. So coming to the next section, what we are going to cover in this is where there are six building blocks of biodata pipeline in Python. So in this we'll cover Python basics, NumPy, Pandas, Biopython, visualization using Matplotlib, and Seaborn for sequence stats. And lastly, we are going to show you how statistics is done by using Python. So first one, Python basics. So as I said earlier, it is a lab assistant in case of a researcher. So what it does, it is a programming language which is used to automate tasks and analyze biological data. So generally what it does is it read DNA sequence files, it calculates sequence lengths, and it also process thousands of gene automatically. So in this video, I'll show you what are variables, loops, function, files in and out. And the second tool in Python is NumPy. So why do we use NumPy? So what it does, it performs fast mathematical and numerical calculations on large data sets. Suppose if you're having a large data set, it performs calculation. So in short, it calculates maybe average DNA sequence length, it finds maximum, minimum, and standard deviation of a sequence length of a DNA. It works efficiently with millions of numerical value. So this is why we are using NumPy, and the third one is Pandas. So basically what Panda does is it organizes biological data into tables. It is kind of similar to Excel, but it makes easier to filter, sort and analyze the data in a set. If we use Panda, we can find out sort of genes by length, sort the genes by length, analyze gene metadata, and so on. And fourthly, it is bio. What I'm going to show you is Biopython. So basically what it does, it's provide ready-made tool working with biological sequence and file formats. Instead of you being writing the complex code, it write behalf of you. So what it does is it read FASTA files, reads gene bank files, calculate GC content, translate DNA into protein, and also along with this it finds the reverse complements in the DNA. When DNA is converted to mRNA, so it finds the reverse complements. So the fifth one is visualizations. For visualization, we are going to use Matplotlib and Seaborn tool. So as humans, we tend to interpret graphs easily when compared with the number data. So it generates bar charts of gene lengths, for example, and histogram showing distribution of sequence lengths, or it is being helpful in plotting line plots of GC content across samples. And lastly, by using Python, we can do statistic tests like mean, median, t-test, and p-value test, p-value, which is crucial in biostat. In this video, I will show you without installing any Python how we can do Python coding. So firstly, go to your browser. So click on Google Colab. Just wait for few seconds. Yes, here we go. You can see the first link, click on it, wait for few seconds. Yes. So after clicking on the link, this is the site. So you can see this is the interface. So we'll start our coding journey here. First click on file, open new notebook. This is the first one, click on it and wait for few seconds. It's loading, let's wait few seconds. Yes, so this is Python, and you can see this is where it is mentioned start coding. So as I said, Python is nothing but if you give an instruction to this, it will do as you say. So we need to learn initially its language. If we are giving an instruction, it will give a result, what we say. So first we'll learn how to communicate with Python. So suppose I will tell him to say hello world, just to display the hello world. What should we do? You see here, start coding. So to tell him to show, first we have to type print, 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 and give bracket and give double quotation. So and type hello world. So this is our instruction. We have given display hello world. This is what we have said. and we have two options. First you can click on this play button, otherwise you can just click shift and enter and we wait few seconds. Hold on a second, just look at this. So we said we have given instruction to the Python print hello world, so it is showing hello world. Just we gave an instruction to display this. So the print is nothing but we are telling him to show or display this word. So before getting into real example, we'll learn some basics about Python and terminology. So in Python, just a minute, we'll remove this code. So first we'll begin with variable. So what does variable mean in Python? So consider variable as a box which stores information. Suppose gene, type gene and gene equals to. So remember one thing, if you're giving a word, it should always start with a quotation in variable. If you give double quotation, it will treat it as a text word. So gene will give as A, T, G, C and GT. So gene is nothing but variable, and in this box we are it has our gene sequence. So what I said, gene is variable, it is box. Inside this box we are storing our data, right? So again we'll take it as length. Length equals to length in gene. Suppose we have given our gene, our variable here, and we are instructing our Python to display our gene. So this is our box. If we print, if we run this, what will happen? The guess is it will display our gene sequence. What is in the box? Just look at this. We got our gene sequence here. Suppose we are instructing to print to display length of our gene, length, th, enter. So length means number of letters in this gene sequence. So there are 6 letters in this, right? So we just gave an instruction. So what is the length of our variable? So it is given as 6. So one more. So this is called as our variable. And for word we are giving in double quotation. What about numbers? In numbers we will never use any quotation. If we want the numbers treated to be as numbers, we are not giving any quotation. If we want our numbers treated as text, we'll give in double quotation, but we don't want that. So gene is equals to 150. And why is that so? I'll give you a simple example. So let's consider A is equals to 150 and B is equals to 10, right? And I will give an instruction to print A plus B. Done, right? So what did we give an instruction? They are telling him to display A plus B. So what will it do? It will add. Just see. A is equals to 150 and B is equals to 10. So 150 plus 10 is equals to 160. Suppose if we give this in double quotation, what will happen? So A is equals to double quotation 150, enter, B equals double quotation 10. So we'll go with the same print A plus B. So shift enter. Just see, this is the difference. In the first code, we have given here without double quotation, so this treated as numbers, and the second one it is treated as a word. So what did it do? It just combined this both. So typically, if you use number, it is called an integer. So suppose if you're using a decimal, it is called as float. So 10.5. So this decimal is in Python, it is called as float, and for number it is called as integer. So there's a difference. And suppose if your code is like any statement, this gene has 150 sequence equals, there's two options, true or false. If your coding has this condition, so this is called as boolean. So there are different terminology for all this. So I hope you understood what is variable in Python basics. So variable is nothing but a box which has data information. So let's go with the next basic thing about Python. It is loops. So these are nothing but tools or instruction. So what basically it does it, this lets you repeat an action without typing it again and again. So basically imagine you have 100 genes names and want to print each one. So you will individually type print and gene. So suppose if we're having genes equals will include number of genes. First will give, consider has one gene BRCA1, right? No, we have to keep in caps lock. BRAC, BRCA1, and second is, don't miss out your quotation or else you will get an error. And the second one is TP53 quotation. We'll go for the next one. We'll take EGFR. Okay, we have done this. So in our variable there are multiple gene sequence, right? So rather than telling to print one at a time, we are telling it to print all at once. So for not here, for for gene in genes print gene. Just a minute, if you get any error, it happens. There is some error. Yes, just with a small deviation we got a huge error. So what we have done is, the symbol which we use here is this. So once you're done, just click on it. See, rather than going for one at a time, we got all at once. So this is loop. Instead of doing repeating it again and again, we can just do it in one single click. So this is our loop. So next we'll go with functions. So we'll remove this loop. So what is function? The next terminology. So function is a machine, or you can say we giving an input, it goes and work happens, and then output comes out. So what should we take an example as? So let's go with define greet gene and name. Then we will tell him to display name is a gene we are studying. So we are giving a function to this. So what did we instruct? So I have used this code. So what does def mean? So we are telling him, we are giving an instruction. So telling him, defining a new function. If we use this, so greet gene is the name we are going to give for our name. So think this has a recipe. We are giving this as a recipe name for greet name. So name, here it goes what we are gonna do in the next step. So basically it is a placeholder. It is just like a blank. So we have to give one thing to display, then it alters this name and gives. Just let me do the next code. So I give an instruction. So greet gene. So we gave our function. Now see what it does. We'll give one gene sequence BRCA1 1. Then shift enter. So let's see greet gene. Then we did give a name. Wait. We did not give a double quotation here. Yes, shift enter. Look at it. So we mentioned it to define a new function having a name of greet name, and we have given our placeholder here. So what we have done, we have given instruction. If suppose this gene name we are including here, so start with this. So it has greeted. BRCA1 is a gene we are studying. So this is how we give an instruction. Suppose if you do a minor mistake in this, we'll get an error, but there is no need to be terrified. It happens, but over time we can manage. So let's go with files, the last terminology in Python basics. So reading and writing information saved on your computers, typically we don't have data written in code, right? We have in Excel or text or file in other and in DNA sequence. So there, in Python specifically for calculation, we are using NumPy as I said earlier, and for plotting, we are using Matplotlib for graphs. And if we are having our data in Excel, we're gonna use Pandas. So separate tools have different use. So one on one we'll go with different tools and I'll show how and what are the terminology used in these tools. So the first tool is NumPy. We'll see about this and what is the use. So NumPy is one of the library in Python used for working with numbers very quickly. Suppose I will show you. Suppose if we have lengths of DNA sequence, right? So what should we do? So to initiate this tool, we have to start with our code. We are giving an instruction. First, how to give instruction to use NumPy. First, we are gonna give import NumPy has np. So we are instructing the Python to import this tool and giving a short name np. So this is what we have given instruction for this. So length equals np. Np is nothing but numpy array. What is array? Array is nothing but a sequence, and we are gonna give in brackets. We'll start with 100. So for numbers we are not gonna use double quotation. 90, and lastly 150. We'll go with this and we'll tell to print its length and the ngth. And so as I said, NumPy is calculated, right? So we'll check our mean of this gene sequence. So this is nothing but gene sequence. In the array is nothing but sequence of numbers. So 100, 120, 90 and 150 is nothing but gene sequence. So suppose if you're having a data of this kind and if you want to find length, suppose if we want to find our mean in this data, so we'll give an instruction print NumPy. We are gonna check for mean of, we have given a variable, right? Length, name of our variable length. So what happens if we start this program? So enter shift enter C. So it has given the length and the mean np name. Suppose if we want to find maximum, print np max length. Just look at it. So given instruction to find out the mean, it has given. So it is nothing but a calculator. So if you want to initiate NumPy tool, you have to give this instruction. So we are telling import our tool NumPy and give it as a short name as np. Right, that's it. So this length, so this is variable equals np. So what we are giving instruction here, so consider this data in np format, in numpy format, and we have given our data in a sequence, and then we have given an instruction, to measure the mean by using numpy. Just look at it. If you click, you'll get numpy, an array object of an arbitrary homogeneous item. So you can check in the Google Colab. So if you use Google Colab, there is no requirement of installing Python separately, right? So this is what NumPy does. It does calculation. We'll go for the next one. We'll go for Pandas. As I said earlier, Pandas are nothing but a tool in Python which are used to for Excel files. So just like Excel, it does work. So for NumPy, we have given an instruction to initiate. So for Pandas also, it goes the same. Suppose if you want to initiate Pandas tool, what we have to do is import Pandas as PD. We are giving this a short form, right? And we'll give our data. First, we'll go here with example. Then later, I will show you an example and how we import that file in our Python and how we can run our tools and analyze or segregate our data. So firstly I will just give you basics. So Pandas, we have taken this. So we'll start here with Pandas. So I have given instruction. So Pandas has been initialized. So let us give our data. So while giving data, be careful. If a small error happens, we'll give in this bracket. So we'll enter our gene. No. Gene. Firstly A, then comma, B, comma, in a sequence, and C, finally C. So data has come, so gene has been done. So we'll take length as well. Length similarly has been done for the gene. We'll take this and we'll keep our data. So 100, let this be 100, 200, and for the C we'll take 150. So we have given our data for Python. For data frame, so this is a data frame. So basically in Pandas, if we are having an Excel data, so we call this as TF or data frame. So we are telling in data frame format PD Pandas data. Yes, it has given automatically data frame for data. So we have given our instruction here. So in Excel format, suppose this is our Excel file. In first column we are having gene and these are the genes, and for length we have given our length here. So we are given an instruction in, so the speed is nothing but Pandas, right? So at lastly we will give an instruction to display this uh, data frame. If it doesn't come, no need to be, yes, this small error. Just hold on a second. Let us see what we did the error. If you get an error like this, don't be afraid. It happens sometimes. The instruction with the, So we have imported pandas as PD and we have given our data. So what did we miss? Yes, so we missed a comma here. Yes, look at this. Just with simple comma, we had an error. So what we have done, we imported this, we have given a gene sequence. So there should be a separation, right? With the gene and with the length. So it will treat as different columns and rows. Just look at here, it has given a serial number and it has given just like a Excel format. So it is just showing us gene and length. So after each section, just give a comma and give your data and show in pandas format. It will show as pandas format, right? So this is it for the pandas. Next we'll go with sequence. I think everyone got this how it works. So we'll remove this code. We'll go for Biopython. So what is Biopython? We'll remove this. So as I said earlier, for separately for biotechnology or for it which is related to biology, there is separate Python tool which is BioPython. So before this, we might have to install a Python BioPython here. So to install, we'll give a command pip install BioPython here. So we'll run. It'll take few seconds. Just wait. Our biopython is started to install. Oh, it is already done. Wait. Collecting biopython downloading. This is the interface which you get once if you install. Basically NumPy and other tools it is pre-installed, but for biopython we might have to install in our Google Colab. So once if you installed, you'll get the interface like this. So installation is done. Once you have installed, you'll get a you'll get a comment like this: you successfully installed biopython. So we'll start with our code, right? So we'll go with a sequence. So biopython is already installed. So how should we begin with? So firstly we have to initiate biopython tool has been done for our pandas and for np. So we'll give a command from bio dot sequence import sequence. Then we'll give our variable DNA in what format? In sequence. So for all other files, did we notice we have given for numpy np, for pandas pd, right? So for here also we'll give our data here. Before that we'll give a command. It should be in biopython format. So we'll give our data here. So suppose we'll take a gene sequence A, T, G, G and CC. So this is our sequence, right? So next we'll tell it to display what? Our DNA. So once it is done, click on it. Just look at this. So we got our file here. So what we are telling it to do is creates a biological DNA sequence. Suppose we want to measure GC content, right? What does GC means? percentage of G and C letters in a sequence, right? So higher GC often means stronger DNA stability, right? So we'll give one more. We'll give an instruction to this and we'll measure the GSC content in this particular sequence. We'll use previous example CC, right? So this is our variable we have given here, and we're gonna give one more instruction. So we are gonna tell it to find out the GC content. So we're gonna give GC. We're gonna give the name DNA count. In that, in particularly, we'll give separately G. Other than G, we want C as well. So we are given an instruction to this. To measure C as well. Finally, we'll give our brackets. Don't forget to give brackets separately, because here we have given for G and C, and whole thing should be covered in brackets if you're giving an instruction to this. And we'll give our code. We are telling this to do with based on length DNA. Then for multiplication we have to go with star symbol, then 100. So we have given this, we are measuring using this formula. We're gonna find out GC content individually, G and C. Then we have given our formula here. Finally, we'll give last instruction to display our GC. It should be in capital, right? No, it's right. Hold on a second. Let us go through once. So we give an instruction here. GC is done. No. Yes. So last time what I have done is I missed this division. Yes. I couldn't click, I guess. Yes, it's done. Just look at this. We have given instruction and division as well. So last time I missed this. So if you miss, you have to be very careful. As we have got our GC content, it's 66.66%. So I think we are cleared with Biopython. I will take one example later and I will show how with this separately, if we have any data in Excel, how we can import in Python, then we can run number of tools and find out what we need. So firstly, we'll take out this. I think clear. We'll remove R. So that was a little demonstration about Biopython. So we'll see about basic terminologies in Biopython. So firstly, sequence. So previously we used sequence, right? So what it does generally, so as I said earlier, it stores and manipulate biological sequences such as DNA, RNA, and protein. Basically, what we used later previously was to store DNA and RNA sequence, and using of this terminology in Python, we can find complements of for guanine and other, right? So basically, this is what it does. Other than this, it translates DNA into protein. So we all know central dogma. This is the scene where DNA translates into mRNA, then later on from mRNA to protein. So in this, you can see this is DNA. So you can see adenine, guanine, cytosine. From here, it is translated to mRNA. There is no uracil in DNA. So by using the sequence or biopython, it finds the complement, reverse complement. Rather than doing manually, it does automatically. And from mRNA to protein, this is the central dogma. And you can see below here, to generate reverse complement, we have to use this instruction in Python, and I have given instruction along with what it does. So if we give this instruction sequence reverse complement, and if we give a variable here, it gives our flip and it gives our complement. The same for 3, it try transcribe and translate. So this is central dogma, right? First it transcribes then translates. So this is what it does. And coming to next, so next is Python sequence IO. So what is this? So this is also basically what it does is it reads and writes biological sequence files like FASTA and GeneBank. So not all the data is in Excel or document. Some of the gene sequence we get in FASTA format as well from GeneBank, right? So this sequence IO, it interprets our FASTA file. So there is one more terminology, parse faster. So parsing means reading information from a faster file. That is what it means. And we'll previously calculate our GC content, what we have done in GC content to find out the percentage of guanine and cytosine in DNA. We used formula previously, right? So this is what we have done. GC content percentage is equals to guanine plus cytosine by total base. Previously what we have used is that the formula into 100. So we measured our DNA stability by calculating GC content by using Python. So this is all about BioPython. Let's go for the fifth tool in BioPython is for visualization. We're gonna demonstrate it in the upcoming example, but before this we'll see basics of Matplotlib and Seaborn. So what is Matplotlib? So Matplotlib is a tool of Python. It is used to create graphs and plots from a data. If suppose you are having a data, by interpreting the data, it automatically creates, if you give an instruction, graphs like this. You can see below. These are the bar graph. Just look at this. Just if you give an instruction from your data, it generates whole graph. I will demonstrate from one example in the later. First let us see this. So Matplotlib, it creates graphs and plots from the data. And what about Seaborn? Seaborn is built on top of Matplotlib and creates more attractive and statistical graphs. So basically, why do we use this visualization tool? For easier plotting and publication quality figure also, it can be done by using this. This is about visualization by using Python. These are the basics of Python. So we'll take one example and see how by using these tools we can interpret, analyze and calculate what is required For our study, firstly we'll take one example. We'll go in our Excel. We'll take a small example. Other than Excel, you can use document file or FASTA and so on. But to show you, firstly we'll take one example from Excel. So we can take i sample id. This is our first information. Here we'll take 5 example here. We'll name it as S1, S2, S3, S4, and lastly S5. So we have given our serial number, so we'll take our species, right? We'll take a species here. Organism, we'll name it as organism. So this is fine. So we'll take a DNA sequence. Just for demonstration I have taken this. You can take whatever you want. By using this file and we using Python, we'll see how all the tools are used and the purpose. And here we'll take gene expression. We'll expand this. So we have given 5 for now. So organism will give human. We'll give here mouse. Again we'll leave it here. We'll give human again. Okay. Rat. And lastly human. Okay. We'll give a short DNA sequence. AT GC GTA CGTA AT guanine cytosine GCTA ATTT GC JT. We'll give this GCGA TATAT. Okay. See TTA just for demonstration. And GNA expression will take in decimal. So as I said, decimal we call this as float, and for number we call it as integer, right? 15.2, 9.8, and lastly 20.1. So we made our example here. So firstly we'll save this. Yes, we'll save in download and we'll take as sample 1. So here we go. We saved our file. Okay, Google Colab is here. Firstly locate, we'll go for new. Wait for few seconds, sometimes it takes a minute. Let's wait. Yes, here we go. So firstly we have a sample, right? So we have to give an instruction to this Google Python to upload our file. So to upload our file, we'll give this from Google Colab import files and uploaded is equals to files dot upload. So simply we're telling we need to upload a file in Google Colab from my file. That's what we are given here. You can either press click here, or you can just simply enter shift enter and wait a few seconds. If you get error, just look at your code once again. There might be a little mistake. Let's wait. Just look at this. So we give an instruction to upload. We got an option choose file. So click on this. So it redirects and opens up my file. So this is sample one. So this is our example. Just for demonstration I have taken this. So click on this and import. Yes, it has imported. Just look at this. So it has given as name sample. It is in Excel format, right? So I said earlier, for Excel format we have to take pandas. So we are gonna begin our pandas. So what should we do? First we have to give instruction to our Python to initiate pandas. So we are gonna tell it has to import pandas has, hold on, pandas has pd, then data frame, so data frame is nothing but Excel sheet. So pd in this format, read our excel file named, so given double quotation. So our Excel file name is sample1 sample. So be careful with capital letters. Sometimes if you just give small, it won't be able to read, because give the exact name of your file, sample1 dot, it's in XLSX format, right? So basically the Excel, it is called a data frame. I told the data frame in pandas, read the Excel sheet, name this. So lastly we are going to tell it to print our data frame, and after that click on it. Look at this. We firstly uploaded our file here, right? Then we have initiated our pandas, then we have given instruction to read what our file is. Look at this, we have got in Excel format, right? It completely read a file. All this instruction has its name, meaning. So firstly import pandas as pd, so it imports panda library. And the second one, pd.read excel, it reads Excel files. And df, it stands for data frame. Basically it's a table in Python. And printdf displays the table, and we got our output here. We have given input here and we have got output here, right? So as I said earlier, variables, loop, functions, and files. So firstly we'll use our NumPy and find out the array. So as I said earlier, cancel this. So we'll import our NumPy here. Import NumPy This is done. So next we are gonna increase our expression. So expression is done, equals np, which is our numpy, and array or sequence. So we'll include in data frame showing our gene expression column. Be careful with the spelling or else you may get error. Expression. And lastly we gonna give it instruction to display expression. So let's see if whether it's right or wrong. As here we go. So in this data we have imported numpy and we have given this array instruction. So we got our array sequence here. And suppose if you want to find the mean of this array, so we'll give another instruction to find out the mean of our gene expression. So we'll give very short instruction. So we're gonna tell it to display print our numpy in mean. To get a mean, type it mean. So we want our expression. So give enter. Just look at this, we got our mean just by using our tool from our data. So suppose you want to find median, similarly as we done for the mean, we'll look for our median. So print median lastly of what? Expression. Then shift enter. So we got a median. Similarly we can do for the maximums as well. So earlier we have done Biopython, so we'll lastly go to our Matplotlib for visualization by using this data. So as I said, Matplotlib is used for graphs, right? So firstly we're gonna import our Matplotlib tool. Then I'll just show you a demonstration. Matplotlib dot pipe lot as, just be careful with the spellings, matplotlib as plt. We have got the option directly as here, right? So we'll remove this. We have got this. So we want our bar graph from this. So we'll give our instructions. So plt dot bar, we need our bar graph. So we'll give our data frame from our excel and simply we're gonna give our sample id. What was our sample id in the first place? So it was sample dot xlx. Just a second. Sample 1. Sample 1. Here we go. So sample 1 is done here. Sample 1 on. Other thing is gene expression. So G in caps and X, it should be in caps. X E X P R E S S I O N. So expression is done. So lastly we have to cover this. So we have given an instruction to import matplotlib as plt. So we have to get in bar graph, right? So we have our data file data frame format. We have given a file name sample1. Then next we want graph of our gene expression. This is done. So we'll draw our x-axis and y-axis. So we'll give our instruction Matplotlib x label is our sample. So did we give sample in capital? Yes. Now we have to give here sample id. Control Z. So sample id. We'll draw sample id versus gene expression. So this versus this, right? So sample id. Okay. And in x-axis we want name sample. So simply what we are given instruction is to plot a graph x-axis y-axis. So sample id versus gene expression, this is what we are going to give. So specifically we are mentioning in x-axis to label the name as sample, and coming to our y-axis, y label, we label y-axis as, we'll go for, just a minute, we'll go for gene expression, gene x, no, we'll just give it expression expressi1. So this is what we are going to give the instruction. So already our plot is drawn. So lastly we are going to tell it to display in Matplotlib, and finally this is what, hold on a second, there might be any changes in this. So we have plotted this, we have kept it in this, we have to give our double quotation over here. So double quotation is done. So bar is given here, our data frame is selected, sample id is selected, and hold on a second, we'll give a title as well. So what we have done is we missed our sample id. So in the file it was small letters, so I have given in capital, that's why we couldn't do this. Finally we got our expression here. Just look at this, based on this information we got the whole graph, right? So next we're gonna use our Seaborn. So firstly we have to import this, just a small minor instruction issue, you'll get an error. Just look at this, just I missed out sample, it was in small letter, I gave it in the capital, so I couldn't get this. That's it. So let's import our Seaborn as SNS. So this is the short form. So we want in box plot data data frame y equals gene expression. So we have given instruction. Let's see if any error. No errors. So if you give the instruction properly, you'll get within a second. Just look at this. Just I given an instruction, and based on this, it has given the whole graph. Firstly bar graph and then Seaborn. So finally we have used our NumPy, Matplotlib, Seaborn, Pandas, Biopython, and lastly we'll perform our statistics. So to import statistics, first we need to give an instruction to import our statistics. So we'll give from Skype import stats. So already we have seen mean, median, right? We'll go for t-test. So based on this, we'll plot lastly a t-test. So we'll go for human and other species and we'll compare. We'll go for t-test. So we'll take firstly human organism and we'll go for gene expression based on this and with others as well. So firstly human is equals to from the data frame of our organism. So let us see whether we have given organism capital or small letter. So it's capital O, then organism. Organism. So this is it. Organism. It is a complicated instruction, but if you keep doing, you will eventually learn. Then human and lastly our gene expression. Gene expression. So this is done. Human in data frame. From data frame we are going to collect organism of human specifically and its gene expression. So let's compare with other species. Others from data frame of other organisms specifically, right? So from organism right? Others equals comparison with human. So finally we'll go with gene expression. Lastly with gene expression. So We are gonna do whole comparison study, so we given our instruction here finally. So tp equals, we need our stats and t-test in human and others. So lastly we're gonna tell it to display our t-test t equals t and print finally. With this instruction, let's see whether our instruction is proper or not. Look at this, we got a t value and p value. Only if you give a proper instruction you'll get the result properly. If you miss a single word you'll get whole error in this. So this is it from Python. And if you are listening here, thank you very much. I hope I have showed how Python works and tools. If you have any query you can mail us at biotech@gmail.com, or else you can contact at this number. Thank you very much.